So in this video, we're going to talk about some reproductive strategies and technologies. So over the next few slides, we're going to go over a few statements, and you can decide whether you agree or disagree with each of those statements. So the first one, if the technology were available, people should be able to clone themselves. Do you agree with that statement, or do you disagree with that statement? The second statement, embryonic stem cells should be allowed to be used for treating diseases such as multiple sclerosis. So do you believe leftover embryos should be allowed to take those cells to develop treatments for different diseases or not? The next statement, transgenic plants are fantastic and can be used to solve the food shortage issues around the world. So a transgenic plant would be something that is containing DNA from other living sources. So we've inserted genes so that maybe they can survive drought better, or maybe they're able to withstand frost, or maybe they'll have longer lasting fruit. Those are all examples of reasons why you might want transgenic plants. Now that we can clone dogs and cats, we will be able to have our beloved pet around forever, just in younger form. Do you agree that that is true, and whether or not you would do that in the first place? Would you decide to clone Fluffy again? And if you did clone Fluffy, would Fluffy be exactly the same as the original Fluffy? It is perfectly ethical to create transgenic animals that have the ability to produce medicine in their milk because it will save human lives. Scientists have actually been trying to develop animals that can produce medications in their milk. So you could just milk the animal and the medication would come out, and then you could purify it and use it as a medicine. Recombinant bacteria can and should be used to produce medications for treating diseases like diabetes. We actually are producing our insulin through this process and have been for many decades. Adult stem cells should be used for cloning, but embryonic stem cells should not. So adult stem cells would be cells taken from your own body and then used to, develop a new organ or tissue. Embryonic stem cells are from discarded or unused embryos. Plants that have been genetically engineered to be herbicide resistant are perfectly safe. Do you believe that or not? So herbicide resistant plants are already being produced. So when you spread the herbicide on your crop, the plant will survive, but the weeds will die. So now we'll get into the note portion of our lesson. So reproductive strategies. The first one is selective breeding. That's the traditional method where plants or animals with desired traits would be bred with one another. So that leads to developing sometimes even new breeds or traits that you want to pass on over and over again. So you would select the best plant and cross-pollinate it with another plant that has the best traits. There are other technologies as well. So in vitro fertilization, the short form is IVF. That's when we take immature eggs and you retrieve them from the female's ovaries and you combine them with sperm in laboratory glassware. So in vitro, in glass is what that stands for. And then you allow the embryos to divide a few times, and then you transfer the embryos back to the female's uterus to develop. So this process is sometimes used if, for instance, one of the parents is not able to conceive. There's some reproductive issues going on, some infertility. Maybe the sperm isn't motile, so it's not able to reach the egg. Maybe the female isn't releasing eggs at the right time. So instead, you kind of help it along. So you are mixing the egg and the sperm in laboratory glassware and then implanting them into the uterus to develop. Another technology is artificial insemination. So you collect the semen, you wash them, and then take the sperm and artificially transfer them to the female. Cloning is making an exact copy of a gene, a cell, or an organism. That part is the part that's sometimes considered unethical right now, like cloning a human being. It's kind of frowned upon currently. There is a lot of cloning that occurs in nature. It happens in many different species through budding, binary fission, vegetative propagation or reproduction, or fragmentation. So we'll take a look at some of these examples. So the first one is budding. So this is a hydra. This is the parent hydra. And off to the side on its body stalk, it has started to create a mini version of itself. So a little bud is forming, and it stays attached to the parent until it's able to break off and survive on its own. And then eventually this would break off. And then you'd have a parent and the baby hydra, and they would be genetically identical because it's just being formed by mitosis, by a bud forming on it. This example here, lots of plants reproduce by vegetative reproduction. So they may either send out a runner, and then a new plant grows there, sets down its own roots, and as soon as it has its own roots and is able to take care of itself, that runner could break off. Lots of plants, you may have done this on your own, where you break off a leaf and you can root it and then put it into a new plant. All of that is cloning because it's the same genetic material in the offspring as in the parent. And then starfish, they're able to reproduce by fragmentation. You can actually break an arm off one starfish, and then it would grow new arms and replace and become a new individual. And then binary fission, that's how cells divide and bacteria reproduce. They literally just divide themselves down the cytoplasm, and you end up with two identical offspring genetically. So gene cloning. So this is when we manipulate DNA to make many copies of a gene in a foreign cell using recombinant DNA technology. So bacteria, as we said, have circular chromosomes, one single one, but they also have smaller structures that are called plasmids, and they're smaller circles of DNA, and you can remove that, and they easily transfer between different bacteria. It's one way of how they can increase their genetic diversity. So what happens is you remove the plasmid from the bacterial cell, and you cut the plasmid with enzymes, and then you can find out of another organism the gene that you want to replicate. So then you insert that gene into the plasmid, stick it all back together. That's called recombinant DNA. And then you insert it back into the bacterial cell, and then whatever that gene is, it will start pumping out the protein that it's coding for. And an example of this happening is human insulin. That's how we produce insulin now, is through recombinant DNA technology. Therapeutic cloning is when we use the stem cells, or undifferentiated cells. You would have learned about stem cells in grade 10. And there's different types of stem cells. There's embryonic stem cells. Those are taken from embryos or even from the umbilical cord. Adult stem cells are somatic cells that still have the ability to differentiate into other cell types like bone marrow, but they can't become anything. So embryonic stem cells, the reason why they're so desired is they have the ability to become almost any cell type in the body by giving it the right instructions, whereas adult stem cells are often already differentiated to a certain type of tissue. And then we can also do something called induced pluripotent stem cells. So there are cells that have differentiated, so maybe they're supposed to be a bone cell or a blood cell, but we can reprogram them to become pluripotent stem cells, meaning that they can be any type of cell with the right instructions. So stem cells can be programmed to specialize and become certain types of cells, and we can use therapeutic cloning in a variety of treatments. You can use them in cancer research and cancer treatment, treating neurological diseases. There's also some steps in treating heart disease and even diabetes. If you can reprogram cells to start producing insulin again and implant them back into the host, the person that has diabetes, maybe they won't have to take insulin anymore because they now have tissue that can produce that themselves. Reproductive cloning is when you take the nucleus out of an egg cell, and then whatever organism you're wanting to clone, you take a nucleus from one of their cells and inject it into the enucleated egg. So we've taken the nucleus out of the egg. You then implant the egg into a recipient womb, and the offspring will be genetically identical to the donor of the nucleus. And this happened with Dolly the sheep. So here's the example of what happened with Dolly. So they took a cell out of a sheep's udder. They took out the nucleus. They fused it with an enucleated egg. So we had taken the nucleus out of the egg cell, put them together. The fused egg they allowed to divide normally to form an embryo. An embryo is a cluster of cells. The embryo was placed into the uterus of a foster sheep, and then the embryo developed normally into a lamb called Dolly. Now, this was the first time we cloned a mammal, and Dolly was fine for a few years, but then Dolly started showing signs of arthritis and other old age symptoms because the donor cell was taken from an adult sheep, and the cells were old already in Dolly. So Dolly actually had to be euthanized early in her life because she was in so much pain from having arthritis and other old age symptoms. Finally, there's transgenic organisms. Those are organisms whose genetic material includes DNA from other species. We call them genetically modified organisms, or GMOs, and there are some examples that have been used. So golden rice, it includes genes from 4 other species to allow for a better yield of rice, but also provides iron and vitamin A, those are 2 nutrient deficiencies that are found in developing countries due to lack of nutrition. So golden rice, the idea would be that it would provide the missing nutrients and provide better yields, so better crop yields. Unfortunately, because golden rice is patented, you can be fined for having some of the seed end up on your farmer's field, even if you didn't plant it yourself, because there's those patent rights. Transgenic pigs, the thought that they would, you could cause them to grow human organs and then be able to use them for organ transplants. Spider silk and milk is a current project of a team of Canadian scientists where you would milk the cow or sheep or whatever animal that you're putting this in, and then you would get spider silk, which is a very strong fiber.